Okay, this question is on the lungs. Pneumonia is a severe lung disease, can interfere with gas exchange. A person with pneumonia can be connected to an ECMO machine, performs gas exchange functions of the lungs. A cannula is inserted into the right atrium, which takes blood to the ECMO machine. In the ECMO machine, blood is passed firstly to an artificial pump and then an oxygenator. So it oxygenates the blood. Where exchanges occur, the blood is then warmed and returned by another cannula back to vena cava. So complete figure 5.1 to show how ECMO machine is connected to the right atrium to the vena cava using a single line to represent each cannula. So this is how we could draw. Okay, so we will have to draw the line, all right, from the right atrium, okay, into the artificial pump, and then back into the vena cava. So this is vena cava, this is the right atrium. All right, so make sure you guys are aware of that. This is also another vena cava, and revision, this is the aorta. How? Because this is the characteristic features of the A shape that we can see here, and then this would be the pulmonary vein. So again, take note, structure of the heart, that's part of your transport in mammals topic requirements. So in the oxygenator, a partially permeable membrane separates the blood from the air that has been enriched with extra oxygen. The name of the structure in the gas exchange system that has the same function as the partially permeable membrane. So the only gas exchange structure in mammals is the alveolus. All right. So this one would be since this is talking about the one with a partially permeable membrane, so that means it is asking about the walls of the alveolus. Walls of the alveoli, because the only gases exchange system in our lungs is the alveoli. All right, so that's where oxygen and carbon dioxide diffuses in and out. In the oxygenator, blood and oxygen-enriched air flow in opposite directions. Suggest and explain how they carry a function of gases exchange. All right, so when it comes to gases exchange, it will be involving diffusion. So meaning it's going to be, so when we need to talk about the diffusion of oxygen and carbon dioxide, and of course whenever diffusion is involved, it has to be linked with how the concentration gradient is maintained, because that's the definition, meaning it's the movement of molecules or substance of molecules get down from higher concentration to a lower concentration down a concentration gradient, right? So the function of this oxygen enriched airflow moving in opposite direction, because it allows oxygen diffuse where? Into the blood, and carbon dioxide to diffuse out of the blood down the concentration gradient, and then that this continuous flow allow for the concentration gradient to be maintained, okay, so that diffusion constantly can occur. Okay, the idea that we recognize the movements of the gases involved, right? Okay, so points of movements, okay, which gases, concentration gradient, and how this concentration gradient is maintained. Next, we have a photomicrograph of the transverse section of the human aorta, okay, the largest artery in our heart, right? And we can see that we have the tunica media, tunica externa, and this would be the tunica intima, all right? Okay, and then this is the lumen of the aorta. Okay, right? So figure 5.2 explain how the structures of tunica media is different from the structure of the tunica media in muscular artery, and relate the differences to the function of the aorta. Now, muscular artery and aorta. Now, please take note, okay, we have either muscular artery or elastic artery. Okay. Okay, now EA stands for elastic artery. The aorta is an example of elastic artery, alright? Okay, I want you to take note of the summary here, alright? But before that, okay, take note what are the structures that can be found in the tunica media. So the tunica media normally contains smooth muscle, right? Elastic fibers and collagen. Whereas the tunica externa, okay, they contain collagen fibers, elastic fibers, and connective tissues. Now, the difference between the elastic artery and the muscular artery is depending on the compositions that we see in their structure. So I want you to take note of this summary table here. Okay, this is the elastic artery and that's for the muscular artery. So the first row here is talking about the position, distance from the heart, right? So the closer it is to the heart the artery is, there tend to be elastic artery. The further away they are from the heart, they tend to be muscular. All right, and then proportions, next, second row, proportions of the tissues that we can find. Okay, elastic artery has more elastic fibers, okay, whereas in the muscular artery it has more smooth muscles. Okay, smooth muscles. Okay, the walls of the elastic artery, okay, the next row, normally thicker. Okay, the walls of muscular artery is thick with more smooth muscle in its layer. And then why is it important? The functions, okay, the functions of the elastic artery is it enables the arteries, because it has a lot of elastic fibers, right? It allows the artery to stretch when there is higher pressure, and then recoil when pressure decreases, right? Why is that important? Because that then will then allow blood movement or blood flow to be maintained. Okay, you're maintaining the volume, the amount of blood flowing through these structures. Whereas in the muscular artery, the main functions of having that smooth muscles, it controls the volume in terms of when vasoconstriction or vasodilation is going to be occurring, because smooth muscles are able to contract, right? Okay, and relax. So when contraction occurs, that's what leads to vasoconstriction. Okay, I hope you remember in your O-level studies the idea of vasoconstriction and vasodilation. Masa you learn about homeostasis, okay, regulation of body temperature, right? Remember that? Okay, so this is the structure that involves in vasoconstriction and vasodilation. Okay, take note. Some biologists, next question, transport carbon dioxide in Cayman laterotris. Okay, and they found out when they respire, most of the carbon dioxide is converted into hydrogen carbonate ions in the red blood cells. Hydrogen carbonate ions combines with hemoglobin in the red blood cells, and they remain combined with hemoglobin until the blood reaches the lungs. So the question is asking us, explain why the physiology of C. laterostris requires carbonic anhydrase, and the biology that we know from our lesson is that carbonic anhydrase involved in carbon dioxide transport. This is the enzyme that catalyzes the conversion of water and carbon dioxide to form carbonic acid. So we know, okay, that's the function. Why? Because after carbonic acid is formed, it will then dissociate into hydrogen ions and hydrogen carbonate ions. So which is what is needed, mentioned here. So that's why my answer is it catalyzes the formation of carbonic acid from carbon dioxide and water, which then dissociates into hydrogen ions and hydrogen carbonate ions. Okay, because we know, okay, this species converts carbon dioxide into hydrogen carbonate ions. And next, explain why the physiology does not require chloride shift. because we know that chloride shift is negatively charged. It is responsible to maintain the charges in the cell, right? Now, why it needs to occur in what we learn? Because in carbon dioxide transport, hydrogen carbon ions is actually transported out, right? So there's a lot of negative ions removed from the red blood cell that makes the red blood cell more and more and more positive. So that means the charges are now changed. To balance that out, right, chloride ion needs to enter so that it turns back into the original charge. But in this species, all right, hydrogen carbonate ion does not leave the cell. It stays inside the hemoglobin. It stays inside the red blood cell. There's no change in charges. So that's why, okay, it stays. Alright, so hydrogen carbon ions does not leave, therefore electrochemical charges or simply the charges, okay, inside the blood is unchanged. That's why. Okay.